Hi, I'm Rick Fontenot. I'm a data scientist at Lantern. I work on our radar, developing our RADR AI platform. I have a multidisciplinary background. My first career was as an electrical engineer. Um, then I transitioned and ended up starting and owning my own business, building modern architecture, primarily homes, but also commercial structures. And then after some life changes, decided to wind that down and got a master's degree in data science, particularly focused on deep learning and some experimental neural network models. And After those studies, was looking for a place to make an impact. And as a cancer survivor, as well as having a lot of cancer in my family, oncology was an area of focus, and I really liked the work that Lantern was doing, and that's what prompted me to reach out and look to join the Lantern team. And I've been with Lantern for about 2 years, um, and it's been exciting building out different models and approaches to help us gain insights with the RADR platform. Yeah, the blood-brain barrier is a selective protective barrier that separates circulating blood from the brain and the central nervous system. It helps ensure that brain remains stable and protected from external influences. Um, it will, by selective, meaning it will allow essential nutrients and gases to pass through this barrier while blocking harmful substances and diseases that can enter the brain. It prevents a wide variety of substances from entering the brain, which help to protect the central nervous system, but specifically the blood-brain barrier blocks the entrance of environmental toxins, heavy metals, dangerous chemicals, bacteria, and viruses. The challenging side, that it means it also blocks a lot of therapeutic agents and drugs. It's estimated that approximately 98% of the drugs are blocked by the blood-brain barrier from entering the brain. And that presents a major hurdle for developing drugs that can treat the brain and central nervous system, in particular cancers. Yes. So the significance of the blood-brain barrier in treating cancers, there's approximately 120 different types of central nervous system and brain cancers, and a majority of them have no effective treatment options. Uh, Often there are cases where you can do an assay and do a study and it will work, but then when it gets into humans, it just can't cross the blood-brain barrier. For instance, with glioblastoma, GBM, there's been no effective single-agent therapies approved in over 17 years, and that is in large part due to the challenge of finding a therapy that can cross the blood-brain barrier. This is a great area of need because the 5-year survival rates of CNS and brain cancers remain really low. Despite advances in medicine, if you can't get the therapy to the brain, it's a challenge. So 5-year survival rates for GBM are only 7%. And it's not the only one. Brain mets, the 5-year survival is around 10%, and ATRT only 32%. So that's one of the reasons that makes the blood-brain barrier so significant in this area is, one, developing a good therapy, but two, it has to be able to get delivered to the disease in the brain. We're going to take a look at this diagram because it's not a simple answer. There's multiple ways that molecules can cross the blood-brain barrier. And this diagram, it shows, and we've got a summary of 5 different ways, but then there's also this counteractive measure, which is efflux pump, which pushes the drugs back out of the barrier, even if they could cross otherwise. We're going to focus on a few of these. First, the paracellular pathway and transport proteins. Those methods of crossing the barrier typically are more with nutrients and not a method by which drugs can cross. The most common way for molecules to cross the blood-brain barrier is the lipophilic diffusion pathway, and we're going to look more at our focus there. There are a couple other ways that are really important. Receptor-mediated pathway across the blood-brain barrier is something. It's trickier, but larger molecules can be passed through with specific receptors, and this is a manner by which peptides and antibodies may cross. Even though they're too large to pass by diffusion, there is a pathway for it to cross. And then lastly, I mentioned earlier this efflux pump. There are a series of proteins, some of the most significant being the P glycoprotein and the breast cancer resistant protein. Drugs that are substrates of these proteins, even if they're small enough and have all the other properties that would typically allow them to pass through the barrier by diffusion, if they're substrates of these proteins, they're subject to this effect of being pumped back out. So when we're studying drugs and say trying to make predictions on which drugs will cross the barrier and which won't, we have to account for both. Does it have the ability to cross by diffusion or a receptor-mediated pathway, but does it have this counteracting effect where it might also get pumped back out and not have sufficient amount crossing the barrier to be effective? We've spent a good amount of time building models that can predict which molecules could cross the barrier. And in particular, we focused on small molecules. It's hard to build a model that can encompass all of those effects, and we talked about previously, meaning drugs that cross by diffusion are going to have different characteristics than the larger molecules that would cross by receptor-mediated pathways. So what on the RADR team, what we've built is a small molecule predictor, defined generally as molecules with a molecular weight less than 500 Dalton. I'll share a little more details about how the workflow and how the model works, but our initial models take the SMILE string, meaning the structure of the molecule, and we generate all of these descriptive features, around 6,000 features. Some simple, such as molecular weight, hydrogen bond donors, and receptors, things are important to crossing by diffusion. But then we have a bunch of other 2D and 3D descriptors, and once we define all of these descriptors of a molecule, we run algorithms and routines to determine which ones are actually important to crossing the blood-brain barrier. And the initial model and workflow that we released was around 94% accurate on a test set of unseen molecules that are hosted on the Therapeutic Data Commons website, and they have a leaderboard, and our models performed very well. They were at the top of the rankings. And we then took all of these trained models so that we could make predictions on new compounds, both our own drugs at Lantern to evaluate, initial assessment. Do we think the drug will cross the blood-brain barrier? And it allows us to then also do screening on large list of other compounds that collaborators may be looking through. It will also allow you to, if you have a drug that seems effective in other studies but does not look like it'll cross the blood-brain barrier, you can then make modifications to the design and have these different entries with little tweaks to the design of the molecule and make predictions again to say, hey, would this small change allow the drug to cross the blood-brain barrier? So that's how the tool was developed initially and has been useful for us. We're also developing our next version of the workflow, and so I thought I'd talk a little bit about uh, what our initial workflow is and what we're developing next, and how that could potentially not only be more accurate but more descriptive in the things that we learn. So here I have a very high level diagram of how our model was built from a workflow. There's more details going on under the hood, but at a high level, this is the process. I mentioned what we're taking is a list of about 10,000 drugs that have been studied, and we know whether it crosses the blood-brain barrier or not. And we are generating these 6,000 descriptive features of the molecule, selecting which ones are important to the blood-brain barrier. And then we're building a series of models, machine learning models, from a simple logistic regression up to a more complex and finely tuned, not just in parameters, but also the architecture and approach in a neural network to look at more of the interactions between some of these descriptive features. And then finally, after we build these series of models, we have a meta-learner ensemble that takes the results from these different methods of predicting whether a drug can cross and blends them together with different weights across these different models. And this ensemble prediction, as I mentioned before, was about 94% accurate on a test set of drugs that we evaluated. Now, if you take that kind of overall workflow, everything in the box here, shown on the left of this slide, and just keep in mind that workflow, the next version we're building, we're going to repeat that multiple times. And what we're doing and finding is all of those descriptive features and putting everything into single models that can explain not only a drug's ability to cross by diffusion, but then it's also its risk of being pumped out through the efflux pumps. It's difficult for a model to just factor in all of those things simultaneously. And so what we're doing now is in this next version we're developing, is we're first looking at a molecule's risk for this efflux pump effect. And so we're taking that same workflow that I mentioned before, and the first thing we're doing is we're training models and making predictions to predict whether the drug will be an inhibitor of PGP or a substrate of PGP. And then we're building, I mentioned earlier, two of the most important proteins in this effect are PGP and BCRP, which is the breast cancer resistant protein. So then we're building a model for BCRP and trying to predict whether the drug is an inhibitor or substrate. And in the future, we are planning to add the next tranche of proteins that affect this efflux pump. It's tricky because there's not a lot of public data out there. So we've meticulously gathered and built data sets to train these models. And once we make a prediction and determination on whether we think this drug is going to be subject to the efflux pump because it's a substrate of one of these key proteins, we're then subdividing all of our training data into drugs that inhibit these key proteins and might have an easier chance to cross by diffusion, and those that may be a substrate of one or multiple of these and are going to have this risk of efflux pump back out of the barrier. Now, just because a drug is subject to this pumping out doesn't mean that it can't still cross in sufficient amounts. So the reason we're building this workflow and to partition them is we're finding that different characteristics and factors of the drugs allow it to cross the barrier based on which one of these subgroups it's in. And so after we subdivide it, we take this whole workflow again and build a blood-brain barrier model for each one of those, and then at the end can reassemble the data set. And what's interesting about this is not only do the initial results show that we will be improving the accuracy, now you have this insight as to one, does it cross? Two, if it doesn't cross, why? And if it is, for instance, a substrate of one of these key proteins, now you have some insight as to, well, yes, it's a small molecule, and the not only the weight, but the other things such as the factors in Lipinski rule of five are all in line, but it's still not crossing. Now you have a clue as to, well, it's because of this efflux pump issue. And if you're evaluating similar drugs or looking at modifications here, that's something to understand first, and how it impacts crossing the blood-brain barrier. Blood-brain barrier models initially, there's one thing is to not only use your own internal data, but leverage as much quality public data that's out there. And I mentioned, we submitted our models and results to the Therapeutic Data Commons leaderboard. Well, TDC is also a really good starting source for public data. So they included, a set of drugs and studies showing whether they cross the barrier or not, and also provide the chemical structure of the molecule. That was a great starting point for us. But then we wanted to increase the number of drugs in our training set. And so we scoured and found some papers on the blood-brain barrier. It was a little more straightforward. There are papers that assemble other papers and other people's data and help. As we moved on to our version 2 and looking to build models on whether a drug is an inhibitor or substrate of a protein, on PGP, it's one that's well characterized, and there's a paper by Brocatelli where he has done this and assembled about 1,000 roughly drugs and the results there, and that's also a good starting point. But then it's probably not the sample size that we really want. And as you move into look into breast cancer resistant protein, there's even less, just large public available data sets. So what we've been doing for both of those is using natural language processing to scrape through papers on PubMed, data tables in PubMed, look for relevant information in those papers discussing this topic and a particular drug. And then we're using a combination to kind of get from this massive amount of papers, bring it down to relevant context. We're using a mixture at this point where we've developed some modules using large language models to help us process that context, the sentence or the paragraph, give it some instruction and examples so that we can produce a data set that says here's the drug name and here's an assay result where it tested to see, is it an inhibitor or substrate? Does it have actual percentage inhibition? Like what fine grain detail can we get and extract? And then once we have that assembled, we're doing manual review, manual annotation to double check those results and ensure that it's as accurate as possible. If there are conflicts between different papers and it's ambiguous, not include that and end up with the most reliable training data set that we can use in the end. On a small scale for us and for a collaborator that may have, a handful of drugs that they're evaluating, candidates that they want to characterize and test. One is this is a quick and inexpensive way to get that initial read on whether you think the drug will cross the blood-brain barrier. On the flip side, a lot of times for a small project like that, people tend to trust and rely more on assays, a test that's done in the lab. And those cost money, but if you have a very small amount of drugs you're looking at, maybe that money is reasonable, and it doesn't take too incredibly long to get results. This is instant with these models. But one of the things we've have found interesting between these and other models is assays aren't foolproof. What were the conditions it was tested under? Did you explore the whole space? What is it reproducible? So having this prediction from a model, if it contradicts, sometimes it sparks a discussion like, well, was this test, was this assay defined across the range? And it might cause you to look again. And in some cases, until these two things are in agreement, do you fully trust the results? But beyond that, on the small scale, really, uh, a lot of the value here is in screening large sets of compounds. And this could be used for drug repurposing, um, drugs that are approved. You can go evaluate across all of these which ones are likely to cross the barrier. If you are looking at candidates for combination in one of your drugs and you're targeting a CNS disease, it's important to know, like, all right, with all of these potential drug combination candidates, which ones do we think will also cross the barrier? There's a whole other slew of reasons, to evaluate the potential synergy of a drug combination, but this is one component that should be accounted for. And then lastly, if you have a promising drug that shows efficacy in a CNS indication, but it's not crossing the barrier when it gets into human, um, are there modifications you can make to the design, small modifications where it can cross the barrier? Now, you'll still have to go back and reevaluate. Does it still have the same activity and behavior? But it allows you to look at a large amount of small modifications to screen through which changes could potentially allow the drug to cross the barrier before, and filter that down into the changes you think could do that part before you start going and doing more tests, not just like a test to see if it truly crosses the barrier, but all of your tests for efficacy and toxicity. So there's a real power in this thing for scalability, just making predictions almost instantly on thousands and thousands, hundreds of thousands of drugs. It's really exciting. I think Lantern in particular, we spend a lot of time uh, looking for therapies for rare cancers. It's underserved, populations of people that don't have a lot of good options, and so it's really exciting in this world of just AI exploding in every industry, every person using it in some aspect on their phone or their computer or TV. Out of all of the possible ways I could take this knowledge I've gained, this experience I get with it, and then go apply it, it's just really exciting to use it in a place that can have this kind of impact.